And now I would like to invite Professor Damjan Osredkar to give insights and preliminary results from the GAIN-CTNNB1, the first clinical trial in CTNNB1 syndrome. You want?
Thank you. And good morning from my side as well. It gives me utmost pleasure to be able to speak about you shortly of the achievements we have made in the last year. These are big milestones that we have achieved, and it gives us pleasure to be able to present this to you. And there's going to be some overlap with the previous 4 talks, which I think is great because some redundancy in medicine is always healthy. But it gives me also the opportunity to kind of glide through some of the initial slides faster. So as a clinician, I actually would prefer that every pregnancy, would result in a healthy human being such as this Botticelli's Venus. But we all carry 20,000 genes, and the nature and evolution actually have a say at how these genes are carried to the next generations. And each of us is born with 50 to 70 new mutations, and sometimes these mutations hit a gene that is actually essential for the human body. So pediatric neurologists often have to treat patients with one of the rare diseases, and one of those, of course, is the CTNNB1 syndrome we're talking about here. I didn't know anything about this disease until I met Urban, and when we diagnosed this disease in him, we started to scramble around in the literature to try to understand the disease and try to see what this brings to him and to his family. But there was not much then at the time that was written. We knew that it was a neurodevelopmental disease, that it's related to a developmental delay with autistic spectrum disorder problems, problems with speech and so forth, but not much more was known. So but the one thing that really intrigues me about the CTNNB1 gene is something that is shown in this particular slide. So cerebral palsy is actually a disease that's much more common in pediatric neurology. It's usually related to problems at birth. But in recent studies, they have shown that actually 1/3 of those patients actually have a monogenic disease that's hidden behind the diagnosis, and meaning that it's the disease that's causing the motor problems that we thought were attributable to the cerebral palsy. And as you can see on this meta-analysis, where they have looked at more than 2,400 patients, CTNNB1 gene is actually one on the topmost gene in this particular meta-analysis, meaning that very likely the disease is very, very severely underdiagnosed, and there must be many more patients around the world that we just don't know of yet. And actually, when we kind of tested this hypothesis in our cohort of patients, where we looked at 135 Slovene patients with cerebral palsy, we found 2 additional patients with CTNNB1 syndrome. So in Slovenia, at the moment, we have 4 patients because there was another young child that was born and we know of. It would be worth, thinking about this fact if we have a successful therapy, because you could move this gene into a newborn screening program and find these patients very early on, and that gives you the opportunity to treat them very early. So when Spila came to me and said we would like to construct gene therapy, I thought that was a very brave idea, but we kind of came on board and said let's see how far we can get. And I think it's no secret here that we wouldn't be sitting here without her courage and also without her energy and perseverance, because without her, none of this would actually have happened. I think what was very important as well was that she has founded the CTNNB1 Foundation, and together with other foundations around the world, this brought patients to the research side, and I think this interplay and discussions that we kind of developed over the years were really crucial in this development as well. Also, I mean, the end money is very important, so the fundraising must be a terrible process, very difficult, and I think Spila knows this best, and also other parents who have been involved in the funding. But fortunately now, as the project is developing, we have also had support from other bodies such as the Sparks NS, for example, who are helping us with developing new aspects of the disease, like trying to find biomarkers that we could use in a clinical setting. But I think networking is really key in this process. Nina has shown you in the previous years about the genotype-phenotype study, and I just want to say that this is how we kind of approach a disease that we did not understand very well, because when we were discussing, the development of treatment for this disease, we were asked questions like, so what aspects of the disease do you want to treat? So which cells do you want to target? And this was not possible unless you understand the disease much better. So the genotype-phenotype study gave us a little bit of insight in that, and we also partnered with the researchers from India, Shubha Tola and Arpan Paicha, who had a mouse model of the CTNNB1 gene. And they look specifically at the brain development. And I think this is particularly important also for the later slides that I'm going to be showing. The CTNNB1 gene is important in guiding the axons of the neurons across the corpus callosum. This is a structure that connects the two brain hemispheres in our brains. But if you have loss of function mutation, if the mice have loss of function mutation, instead of crossing over the corpus callosum, these neurons clump in this area. They don't make the connections. And this kind of proves that the way that the brain is formed in many of these children might be affected. And this was actually also the case in some of the patients that we have met during the years. You can see that the corpus callosum, this whitish structure here, is thinner in some parts. And those white matter lesions are actually quite important, as you will see later on in the slides. We also didn't know whether this is a progressive disease or if it is, a static disease. And many parents have reported in the genotype-phenotype study that about a third of parents reported actually that the disease is progressive. But we wanted to have a more, other clues about that. And what we did is the EEGs that the families provided of the patients, well, the babies and children who were living with the CTNNB1 syndrome, we have compared those to healthy children of the same age and sex. And what you can see is that in healthy children, the two hemispheres, they communicate with each other quite normally. The darker the squares, the more communication there is between the hemispheres, whereas in the CTNNB1 patients, this communication is less abundant. It's present, but less abundant. And what you can also see in this second graph is that with time, the synchronicity between the two hemispheres increases in healthy children, whereas in the CTNNB1 children, it kind of goes down, meaning that very likely, looking at the EEG aspect at least, the disease is progressive to some extent. So we wanted to understand this time development of the disease in more detail. And, we've been hearing yesterday from Nina about the natural history study. This is a really crucial study to carry out in any disease, really, where you want to treat with disease-modifying treatment, because it gives you a background to which you can compare the treated patients, so you see whether the treatment is actually effective or not. And I think it has been a lot of work from everyone involved, from parents bringing their kids around the world to the site, and also from everyone involved clinically, the physios, the psychologists, physicians, because it can be quite challenging to examine kids in hotel rooms and so. But it's been also very electrifying and eye-opening, because until the first kind of evaluation, I've only known Urban, but then on the first evaluation, I've seen 84 kids with CTNNB1 syndrome, and I think that really kind of showed me that many of the kids would have, of course, their specific features, individual features, but then they would carry some common features, and that kind of gives you a good insight in how, this syndrome expresses itself in a child carrying, a gene that's affected. We have heard very elegant lectures about gene development, but let me just underline what Leszek said. It's really, really crucial that preclinical and clinical scientists not just collaborate, but also talk to each other. I must say personally that it gives me a lot of confidence that I not just understand it better through all the discussions that we had, because I didn't know much about gene development in the past. I still don't know many things, but at least I know more than I did before. But also, my concerns and my thoughts, they were kind of incorporated in the development of the therapy. And actually, for example, when we were discussing whether to go into cisterna magna or intraventricularly, we thought that we have to go into the ventricles to reach the deeper layers of the brain, which otherwise wouldn't be approached. And that all was taken into account, and that's how we came to the final kind of protocol. And it gives me confidence, because I understand the treatment a little bit better. I understand what it does. I understand, why we decided for certain features, so I can, give it with more confidence to our patients. So, it took us about, Spila said, 5 years and 5 million euros. So it's a big effort on the side of everyone. Viralhin has produced the GMP product. And we received the orphan drug designation, which is also an important milestone because it gives some confidence that even the legislative bodies are recognizing the need for a development of such a drug, but also that the process through which we were pushing the development was of good enough quality that even the legislators would give their check mark. That we can go into the first in human trial. And we got the approval from EMA in November 2025. And there's currently one site at which we dose the patients, but we are definitely looking to expand it to make it easier for the parents, but also, for example, those who don't speak English might be better off if they speak Spanish to be dosed in a Spanish speaking site. So we have 3 science committees. There's a medical steering committee where we discuss, how to proceed with certain steps in the trial. If there's any problems in the development of that, we kind of take those decisions there. We also have an independent drug safety monitoring board who is there. They are completely independent. We report to them, and they have an oversight of what's happening in the trials. And they're the ones who give the green light, after the first patient was dosed, they gave the green light. There's no safety signals that we should be worried about, so you can go and dose the next patient. And they are taking care that, everything is carried out in a safe enough manner, and that there's no signals that should be worrisome. But we also decided to make an independent ethics committee for the trial site, because maybe one of the most difficult, processes that we had to go through was the patient selection. There's been over 110 families that have expressed an interest to participate in the trial, and we only had 12 doses in this first trial that we are now carrying out. And of course, we knew that we will not be able to help most of the children really that have whose families have actually expressed an interest to participate in the trial. So we had to construct a set of rules by which we would kind of decide whom to treat and whom not to treat. Besides the inclusion and exclusion criteria, we have prioritized those who were participating in the natural history study not because we knew them, but because there's data that shows then how the patients would progress over time, and then by being treated, how this the curve would change if it would change. And that's why. And also, we had to look for patients and families who had realistic expectations, but also those who actually understood the risks of stepping into unknown. Going from preclinical trials to a clinical trial is always carries a risk, even if the preclinical data can be super fine and showing that everything is safe and no problems have been occurring. There has been reports in the past, when they went to the human trials, things could happen that were unexpected, and going to first in human trial is always a risk. So we have prioritized the families who were keener on acknowledging that risk, but also accepting that, so they would understand, that things might happen that we were not expecting. This is kind of a slide because yesterday people were asking about the inclusion and exclusion criteria. I added this slide. I think there's like 10 inclusion criteria and over 30 exclusion criteria. You can find them on the clinicaltrials.gov page. But just to kind of summarize it here for you, there's an age requirement. Patients aged from 2 to 12 years were included in the trial. Maybe in the future this is going to open up. They had to have a stable weight. They shouldn't be like having other medical problems that could contribute to the, in fact, the way children thrive in the trial. The family had to commit really to being in Slovenia for 5 months at least, during the trial process, because they need to be very close to the hospital if anything goes wrong. Of course, they had to sign the consent. There were a lot of exclusion criteria, as I mentioned, like an active infection. Actually, we had to postpone one of the patients because, fortunately enough, in the pre-screening period, we found that there was some potential infectious process happening, and we had to take care of that. And also, if there are any major concerns regarding the patient's health from other organ systems, we had to exclude those patients. Once we engage with the family, we form a team, and I think we were very lucky with the first two families. They were really on board with the trial, and we can communicate very openly about everything happening, because it's not just us telling the families what they need to do and how they need to fulfill all the requirements, but it's also very important that whatever they find in their child or worries them, they need to communicate that with us, because that's how we can actually manage that and treat that as soon as possible to prevent any major detrimental effects that could happen and could be prevented. So, for example, the most important thing would be to prevent an concomitant infection. Having another infection in other trials has proven to be fatal, so we really try to do everything possible to isolate the patient, to keep them from people who are sick, and so that it can always happen, but as much as we can do, as humanly do, it's necessary to take care of. The parents also have to write whatever drugs they were giving to their child, and also if there were any side effects, they had to report that. Also keeping a seizure diary and so forth. So it is some work from the parents as well, and also there's a lot of energy that is taking them just to come to the trial site and to communicate all this with us. So this is when it came to the trial, this is like kind of a very brief summary of the trial itself. It is a clinical trial of phase 1/2, meaning that it's firstly about the safety of the patients, but we also are looking for efficacy. So if we can tease out the efficacy, that's great, but it's not the primary outcome that we're looking for in this trial. That's why this is going to happen in the next kind of phases of the trials, where we're going to design it more specifically to tease that out in a very objective manner. But safety is what concerns us the most. There's going to be 12 patients enrolled. First, we treat the older ones from 6 to 12 years. So after the first patient was treated, we had to wait for 3 months to kind of observe for the possible side effects. Then the data safety monitoring board, they reviewed that and gave us the green light to proceed, and then we could treat the next patient. After 6 weeks of being treated, we can now proceed to the next patient and so forth. After that, the older patients can be treated much faster, but also the part B opens up, where we're gonna go through a similar process with these weights, just for the safety reasons. All the patients will receive therapy, so there's no placebo group in this kind of open label trial. They all get biventricular injections, and there's a dual immunosuppressive therapy that these patients are required. Duško has already shown that, that this is kind of necessary for from the previous trials that are shown, but because there's a massive immune response of the body towards the virus that we introduced in the body, and we need to make sure that this doesn't get out of control. And I know there's a lot of discussion among parents regarding the prednisolone and sirolimus, and we can take that into the open table that we're going to have after the talks. But I think I'll be able to show you that clinicians are very cautious, because our main goal is to carry these children safely through the trial. And there's some signals why we think those, even in our trial, showing that those treatments are important, but maybe we can fine-tune them to a better, extent or dosage. So the first patient was treated in December of 2025. The second one was treated in May 2026, and we think that the next patient might be treated end of August, beginning of September. I'm going to walk you through a few slides. This is Urban, the first patient. In his room, we have sterilized the room before he came in. The parents we all started wearing masks and were very careful of not letting anyone that didn't need to meet him to be in his presence. And this is just before he left his room, where he was then hospitalized for the next week, towards the operating room. And once he was in the operating room, we anesthetized him, intubated him, and then they have prepared Urban. On the left, this white box here is the neuronavigation Peter has mentioned, and some electrodes through which we can actually, which help us then to navigate the tools of a neurosurgeon directly into the ventricle. And this is how they were carrying that tool in the hand, but then looking on the monitor to see if they can follow the trajectories very precisely. And that's how Peter was able to reach the ventricles and deliver the medicine. And this is how the medicine was delivered, following a precise protocol. It took, at some point, there were 15 people in the operating room, bringing the medicine, taking care of Urban, doing the surgery, just observing as myself. But it was a very busy room, and everybody had to know what they were doing and, to do it accordingly to the protocol. But just after the surgery, we were able to wake Urban up, extubate him. And this is him in the intensive care unit, where he stayed for the next day. I was having him monitored every 15 minutes. I would do a neurological examination. And then after a few hours, this was kind of more relaxed. So he could be checked on an hourly basis and then on a 4-hourly basis. And the next day, he was moved to the room from which he came. And this is Mafalda, the second patient, who was, this is how she came from the intensive care unit to her room. So we did observe some adverse events. I must say that the only severe adverse event that happened was actually when Urban kind of scratched open his wound. So he kind of bled from the surgical wound, but fortunately, this was very shortly communicated with us. We were able to arrange, hospitalization. He was resutured. He got some antibiotics, and that was actually taken care of. So it was not the most worrisome side effects that we have experienced. He had a fever for 1 day, which occurred on day 4, which is kind of normal for AV9 therapy. But then on day 7, he started to show some autoaggressive behavior. And this was something that at first we didn't took too much into account, because we knew that steroids could cause this kind of behavior. But then this behavior worsened, and at the worst time, he hit himself, for example, 100 times per day, as the parents have reported to us. And this has been, we thought that it was caused by steroids, but we didn't know. So that's why we did the additional examinations, like additional MRIs and lumbar punctures. But fortunately, when these steroids were tapered off, this behavior completely vanished. So we still think that it was really attributed. It can be attributed to steroid use. We have seen also dyslipidemia, so cholesterol was a little bit higher. This is a normal bodily response to inflammation. And then because of the behavior, we did a control MRI, and we saw a lesion that formed that we didn't see before in the previous MRI. I'll talk about this on the next slide. And then later on day 55, when we did a control MRI and a control lumbar puncture, we saw for the first time really that there was increase in protein in the cerebral spinal fluid, which can be a sign of inflammation as well. No leukocytes were present before or after. And also on the later lumbar puncture, the proteins kind of disappeared. So there was a signal at around 2 months that something was happening, but then that it went away. But on the at around 3rd month, after treatment, we also saw interstitial synthesis of immunoglobulins, meaning that there was some sort of inflammatory response still happening there that was not present before, and showing that this is an active process. It's not just giving the therapy, but the patients have to be monitored really precisely over the next months, and that's also why we think that the immunomodulatory therapy is necessary. So Urban had, like many patients, we've looked at the MRIs, and approximately 3/4 of the patients in one cohort that we were looking at had some sort of a non-specific white matter lesion. Urban had a small perivascular lesion here before the treatment, and we thought that's something that we quite often see in kids, and we don't usually take that into much consideration. But then 2 months after he was treated, we saw that around this lesion, we did this control MRI because of his behavior. We saw that the lesion kind of increased, so there was a kind of a new lesion that formed. There was no contrast enhancement. It seemed like an inflammatory lesion. And then on another MRI, we didn't see any new lesions forming, but there was kind of cystic transformation of that particular lesion. It didn't cause him any additional problems. He didn't develop epilepsy, for example, or any signs of a mass. But it was something that we were a bit worried about, and we thought that we need to keep under control. We'll do an additional MRI next week, so we will know better in how this thing is developing. This is something, this is not from any of our CTNNB1 patients, but this is from another trial. This is the mucopolysaccharidosis type 3A, they also treat patients with AV vector, but a different serotype. And what you see here is that in the ejection sites, lesions like this would form at around 3 months, and then they would develop for several months, but then they would kind of stabilize. And they argued in this particular paper that's already published that this might be a local immune response, or it's like enzyme overexpression that's causing this kind of lesions in the brain. So in the second patient, we didn't see any major, severe, life-threatening adverse events. There's no acute liver failure, like in almost every patient has that when treated with Zolgensma, for example. But what we did see is that on day 6, she developed orofacial dyskinesia. I have a video on the next slide. So we discontinued one of the medicines that she was receiving before that. When she was dismissed home, we also saw that there was an increased frequency of impulsive outbursts like tantrums. I think Bruno will be able to describe that much better on his talk. And we have kind of at least cut this major, peaks of excitement with risperidone, but still this is, again, I think related to steroid use. But hopefully when she will be discontinued with steroids, she will be fine as well. And the orofacial dyskinesia have just recently went away. So in about a month, this was a thing of the past. She also had some sleep problems, but that was easily tackled with some of the medicines. So maybe. Okay, so this is what occurred in Mafalda. After 6 days of therapy, she would have some problems with movement around her mouth. We didn't control MRI in her. We didn't see any worrisome signals. There were no lesions that would be forming in her brain. We might need to do a control MRI before the scheduled one, but we'll see. We'll have to discuss this with the parents and also with the team whether that's necessary or not. As I said, this kind of went away after a month. We're also trying to understand what's happening regarding the post-treatment. Neurofilaments are parts of cells. It's like the poles of the tent. So if there's a storm and the tents are broken, then you would see the poles lying around. So this is something that's commonly elevated after an AAV9 therapy. So we see that some of the neurons are actually dying, but that's like a sacrifice that we are kind of we are okay with, just showing that we do see the increase, but then after, like 2 months after treatment, this kind of goes down, showing that the brain is kind of recovering after this kind of treatment. But what was a bit worrisome, and we don't understand it completely yet, but we're doing more tests on this topic, is that tumor necrosis factor alpha, which is an inflammatory marker, a very unspecific one, really is kind of coming up about 60 days post-treatment, in Mafalda a little bit earlier maybe, but still not as high as in Urban. Whereas in the cerebrospinal fluid, we didn't see this increase. And other inflammatory markers such as interleukin-6 were normal in cerebrospinal fluid and in serum. And the ELISpot test, which kind of shows whether the body is responding to the AAV9 capsule, was also negative. So we have to understand where this tumor necrosis factor alpha increase in cytokines is coming from, but maybe it's just a non-specific response, which we would actually think would happen earlier on, just after treatment, but maybe this kind of explains that there's still a little bit, we have to be cautious on this side. So if kind of summarize all these side effects, many were kind of tackled, they resolved or were managed, but there's the lesion that we are still observing. We don't completely understand what the protein that we found in the cerebrospinal fluid means, but because that went away, and we are monitoring this, and also the interstitial synthesis of immunoglobulins is something that we are kind of monitoring. But as I said, we're trying to tease out also some efficacy, and you've seen this video now a couple of times. This is Urban walking before treatment. He had to be supported really heavily. I mean, he would collapse after one step if he would be just held by his hands, and you had to carry his weight for him to be able to walk as he did. But then, this is also a video that you've seen. This was incredible a video that was sent from the parents where he would do his first steps, independent steps, which was not possible before. He couldn't do even one step. And I think what, to me, as a clinician, was even more impressive than that was the fact that they could take Urban to the zoo, and he could walk for half an hour just by holding, one of the parents for one hand, with his one hand. So I think that shows that he has more stamina in walking, and both of the parents also report that they have kind of hunger for walking, and which is really great, and they enjoy that much more. So Spela has showed this image as well. There was proper evaluation using standardized scales, Bailey scales. And what we can see here is that, particularly in the motor, side of development, we do see an important increase in their Urban's capabilities. Mafalda is too early after treatment to have that. And this slide, I think, also is really underlining what the previous one is showing, and Spela has showed that also. Urban was not developing for many years, but after treatment, he gained 2 months in about 6 months, which I think is a tremendous improvement according to the baseline. And I think he's not done yet, so he's going to improve further. And the walking part, we try to we objectively quantify that, and fortunately Urban was carrying the side device. It's a wearable device like a watch, but you carry this around your ankles. And this has been proven in studies in other diseases that it can objectively measure the quality and the speed of movement. And what you can see here is that he's doing, he would be doing like around 2,000 steps before treatment, but then now he can do 17,000 steps, which again is really impressive. And this was shown yesterday by Spela and Nina. The fact that he can walk is actually something that we did not expect if he was untreated. Bruno will talk more about Mafalda's progress, but I think everything cannot be measured with scales, but improvements like this, the fact that she can actually sit by herself on a toilet is something that it can be a major benefit for the family. And as you can see, she's enjoying herself. And I think that's I think that's a very important aspect of seeing the the effects of treatment as well. And what Bruno has told me yesterday is that Mafalda now also can do a few steps being supported like here, but in the past things like that were just not possible. So we hope that we will be able to see an improvement in her motor abilities as well. So long term, I'm almost up. I mean, I'm sorry for taking more time than was allocated for me, but we know that this is not a cure. So children who will be treated will not become completely healthy children, after treatment at all. But it is a therapy, so we do hope that we will be able to modify some of the aspects of the disease to lessen the burden for the family and for the child. And I think we do see some clues that this might be happening. What we hope for is that the disease would be stabilized, it would not progress, and so the patients wouldn't lose the ability of walking, for example, when they become adults. Now, we hope for some functional preservation, so whatever these kids are already capable of doing, they wouldn't lose. We also hope there are going to be some behavioral benefits. So, you know, if the behaviors that are difficult at the moment could be, you know, melted away to some extent, that would be a big benefit for the families as well. Many of the families on the forums that you're signing here show that behavioral problems are a big issue in CTNNB1 kids, and if we could get that improved, that is really good. And of course, there's a potential to achieve new milestones. So there's a clinical trial going on. We definitely try to expand it to new sites and trying to bring not just 12 kids, but more kids to, towards treatment. And until now, we have seen no severe adverse events that would be worrisome to us or to the DSMB board. But we have to be cautious about any kind of conclusions because there's only 2 patients that have been treated so far. So, you know, things could still happen in the 5th or the 10th patient. So there's still caution that's necessary. So we are kind of reservedly optimistic regarding the efficacy, and we'll see how this develops when we have more data points. And what I think we can conclude also is that, you know, development of a gene augmentation therapy as this one is possible through a parent-led initiative and you know, within a network together with researchers from all fields. And as everybody here in this room, I think, understands by now, collaboration and trust are key. So thank you very much for collaborating with us, and looking forward to the questions in the last session. Thank you. There's, of course, a team that's. Can you just give the last slide, please? That's important. It's not; these are not just words, but I just want to show that there's a huge team at the University Medical Center. There's more than 30 professionals. Just if you could go to the So. So there's, you know, there's the Leszek-Lisowski group, the National Institute of Chemistry group that is helping tremendously. Of course, the CTNNB1 Foundation and foundations, but we have the steering committee, the Data Safety Monitoring Board, and the edX group that is kind of having oversight on the whole process. So thank you all for being part of this group. Thank you. Thank you. Yeah, thank you.